In this video, we have to demonstrate the DNA extraction from blood using the Kaijin and blood minikit. The steps are started by the DNA extractions of a single blood sample. The first step is to collect the sample from experienced phlebotomist. First, we have to spray the bench pipettes and equipments with the freshly made 10% bleach solution and sit for 15 to 30 minutes, then wipe away any residual bleach. Rinse with clean water and spray with the 70% ethanol. Gloves are to be worn at all times. And check that all equipment is in place. Buffers should be prepared for the further processes. Heating incubation will be 50 degrees centigrade and will be ready to use. The second step after that is that we have to label microtubes first of all with sample IDs and label them with permanent markers. Carefully. Now we have to mix blood at least 10 times. Now vertex the proteinase kinase enzymes and followed by the quick centrifugation step by 10 seconds. Put 20 microliter of proteinase kinase in each microtube. Prepare 200 microliter of blood in each tube. Do it slowly as the blood is viscous. Now add the 200 microliter of AWI buffer to each sample. Now close the tube and vertex for 15 seconds and transfer the microtubes to the heating blocks at 6 degrees centigrade for 10 minutes incubation. Now remove the sample from the heating blocks and centrifuge it to remove any droplets from tubes. And now add the 200 microliter of absolute ethanol to each microtube. Mix each sample for about 15 seconds. Now prepare the mini spin columns for each sample. Proper label it and label the spin columns in ensure with proper sample IDs. We usually label side of the spin column. Now carefully transfer the mixture from the previous step to the spin column. Take care and this is repeated for the subsequent samples. Now close the spin column and centrifuge at 6000 gravitation or 8000 rpm for 1 minute. At the end of the centrifugation period, add all of the filtrate into the collection tube and transfer the spin column which have DNA to collection tubes having the lysate. Carefully open the spin column and add the 500 microliter of AWI buffer with carefully. Remember to change the tips every time. Now close the cups and return the samples to centrifuge for 1 minute at 6,000 gravitation or 8,000 revolution per minute. At the end of the centrifugation, check the fill rate, and if there is further need, centrifuge it again. Now discard the collection tubes and transfer the spin columns into the clean collection tubes and open the lid and add the 500 microliter of AW2 buffer carefully in it. Now close the cups and centrifuge at full speed at 2000 gravitation around 14,000 revolution per minute. Discard this filtrate from the collection tubes into the waste containers as the collection tubes can be reused. Dry the collection tube with the paper towel to dry and place the spin column. Now centrifuge at a full speed for 1 minute at 2000 gravitation or 14,000 revolution per minute. Now label the microtube, one for each sample and for the spin column to each corresponding microtube. Now add the 200 microliter of AE buffer to each spin column. Change tip for each sample every time. Incubate for 1 minute at room temperature and centrifuge at 6,000 revolution per minute for 1 minute. Transfer filtrate to spin column and repeat the incubation and centrifugation step. This optimizes the DNA for each step. Repeat this incubation and centrifugation step and this optimizes the DNA for each step. After this step, the DNA extraction of the blood samples is almost complete. Now perform the couples of QC step. We recommend to remove and allocate of each sample for QC. Total of 5 microliter of this is sufficient and leaving rest of your sample in this. Use the spectrophotometer to determine the DNA and purify it. Followed by the gel electrophoresis to establish the DNA integrity. We take the 1.5 microliter of DNA sample and use AI buffer to blank the instrument. A controlled DNA sample is always included of known concentration and we have to use instrument at optimistic level. In accessing the DNA by the concentration by 260 to 280 ratio is important. 260 to 230 ratio is good. 260 to 230 ratio is important in all of this mechanism. Here we can see a right and a wrong result. The next step is to run sample in 1% agarose gel. Leading buffer is added to each sample. Included the DNA ladder which is used alongside the sample. The buffer is added to the tank and gel is included into the gel tank and sample together with the DNA ladder load onto the gel. Gel is then treated with the 100 volt for 20 to 30 minutes. Now gel is removed for gel tank and DNA is now visualized by the patron